in case of the cork cambium, we discussed they present toward the outer side, and basically cork cambium they cut toward the outer side, they form a cork cell, and cork cell also called as the phellum. That same similar cork cambium they cut toward the inner side, and now they form a secondary cortex, and secondary cortex also called as the phallodome. So quickly we are taking the basic difference between the cork cells and cork cell also called as the phellum with secondary cortex, and secondary cortex also called as phallodome. Okay, chalo. First one, cork cell, basically these are the dead cell. First point, dead cell. But when you compare the secondary cortex, these are the living cell. Okay, now obviously we are talking about the dead cell. So dead cell nothing but these are the cell where deposition of some material, there is a deposition of some material. First one,
Excuse me, ma'am.
material, yeah.
Ma'am, in the portion there is figure 8.13 and in the textbook it is not there.
Text me after class. Okay, I'll see.
Okay.
Okay, text me. I think I remember 8.1. I think that is a chromosome structure. जो last वाला point है ना, centromere, telomere, acrocentric chromosome, telocentric, submetacentric. I think that those diagram. Because Malakai shared me the images, so I just check that same diagram actually. Okay, let's see. Yeah, share me after class. We are talking about the material deposition, that is suberin. So in case of the dead cell, in case of the cork cell, first of all there is a deposition of the suberin, and such suberin deposition it is totally absent with respect to secondary cortex. Another one, now there is a deposition of the another material that is called as the tannin. So tannin also present, but such type of the deposition of the tannin, it is totally absent with respect to cortex cell, with respect to phallodome. Now next part, when we are talk about, that is cells. So obviously due to deposition of the different suberin material, due to tannin material, so ultimately whatever the color, so color of cork cell basically they obtain the dark color due to deposition of the material. While when you compare the secondary cortex, so secondary cortex basically these are the living tissue, obviously they are made up by the parenchyma, and now they are lighter in color because there is no deposition. And finally we are talking about function. Obviously dead cell say, and you know very well, is this any dead cell? So major function, cork cell, they are responsible for, they provide the mechanical support, while secondary cortex parenchyma tissue hai, living hai, so ultimately the function, they are responsible for storage of food storage. That's it. So these are the exactly difference between the cork cell and the secondary cortex. Cork cambium, they cut toward the outer side, they form cork cell also called as the phellum. Cork cambium, they cut toward the inner side, they form the secondary cortex, and secondary cortex also called as the phallodome. And exactly these are the difference between the cork cell and secondary cortex. Okay? मलाकाय ऑलरेडी सिलेबस में मैंने शेयर किया हुआ है. Let me, I'll share that same syllabus for you guys also. Just wait. No, ma'am. Okay, so chalo, write down the diagram. Which diagram it is coming from the exam? Please write down. Diagram 2.6, 3.2, 3.7, 5.22, 5.23, 6.4, 6.5, 8.4, and 8.13. Out of this, any diagram, 2 or 3 diagram will become. Now you can check and tell me. Acha.
Diagram for 2 marks, right?
Diagram for the 2 marks, yes. It's for 2 marks. Done? I think 2 or 3 diagrams will be asked.
Excuse me, ma'am.
Hmm.
The diagrams will only come of botany and or zoology, yeah, both.
It's for botany, dear. It's for botany. Okay? And remember, I think ये जो 5.2 and 5.23 ना, these are related to morphology chapter. Just check once. Because second chapter biological classification, third chapter plant kingdom. I think fourth is animal kingdom. Then fifth is morphology. Yeah. So if diagram for the morphology, you have to please remove those part, okay? Because I remove the morphology chapter, so ultimately no need to practice diagram also. Okay? Yeah. So these are the part for your botany diagram. So I'm telling you 2.6 means biological classification chapter, 3.2 and 3.7 from the chapter plant kingdom. Now 6.4 and the 6.5, these are the for the anatomy, and 8.4 and 8.13, that these are the chapter for cell. Okay, clear? Clear? Chalo. So next part, yeah, don't worry. At the end of the class, I'll share detailed message with syllabus and the diagram also. So next part, we are talking about one question. Yeah, differentiation part is over. Now we are talking about one question. Now, basically, we are taking one plant, that is oak plant. Oak plant, and the botanical name for the oak, Quercus suber. Q suber. Quercus suber. Basically, from the oak plant, mostly such oak plant basically it is used for the formation of the bottle stopper. So you know very well, in case of the chemistry lab, basically, for example, is this any concentrated HCl or is this any concentrated chemical is present? So might be possible there is a umber color bottle, brown color, and now there is a something, a wooden cap is placed. Even for lots of liquor also they are having that wooden cap like structure. So that wooden nothing but they obtain from the cork part. Okay, and please remember which part is used. Basically, cork cell part is used. Cork cell. So cork cell it is used as a bottle stopper. And which plant is get used? That is yes, Harap. Cork one. So cork cell basically they are used and they obtained from the oak plant. One question. Okay? Chalo, done? Done? Now we are taking the one another important point, lenticel, which is associated with the cork cell. Yeah. Should I continue? Now, next part we are talking about lenticels. Basically, when we are talking about the lenticell, these are the cells which is always open. First point. These are the cell which is always open. Another one, lenticell, basically they consist of, they having a small pore. They are having a small pore like a stomata, because stomata also they are having the small pore. क्या हुआ?
Ma'am, could you please scroll up a bit?
So we are talking about the lenticell. Lenticell, basically these are the specific type of the cell. Those are having a small pore. Due to that property, we can say these are the always open. Now, when we are talking about lenticell basically they present on stem, as well as they present on root, as well as they present on the fruit. So such kind of the small pore they present on root cells, they present on the stem cells, as well as they present on fruit. But please remember, they are totally absent on leaf. Lenticell, it is not present on the leaf, because on leaf stomata is present. Because on leaf stomata is present. You know very well, in case of the dicot stomata, there is a presence of kidney shaped stomata, while in case of the monocot, dumbbell shaped stomata is present. So lenticell, these are the specific type of the cell. Those are having the very small pore, which is always open, mostly present on the stem region, root region, as well as the fruit region. But please remember, on the leaf structure they totally absent, because already on the leaf small pore is present. That small pore nothing but that is called as the stomata. Now we are talking about what is the exactly functions of lenticell. So lenticell nothing but these are the small pore, right? So whatever the function performed by this stomata, all this function performed by the lenticell. So you know very well, first of all, stomata they are responsible for the gaseous exchange. So similarly, lenticell also they are performing the same function. They are responsible for exchange of gas. So they are responsible for the gaseous exchange. And the another one, stomata responsible for the transpiration. Similarly, lenticels also they are responsible for the transpiration. Question: in morphology chapter, again we discuss the lenticell part, because lenticell they play very important role during the gaseous exchange. So please remember, stomata and the lenticell they are functionally similar. Functionally, because stomata also they are responsible for the transpiration, they are also responsible for the gaseous exchange. Similarly, lenticells also they are responsible for the gaseous exchange and the transpiration. But please make sure, gaseous exchange, basically these are the major function with respect to lenticell, which is always open. Got it? Lenticell, these are the specific type of the cell which is present on stem, root, fruit, always open, small pore, responsible for gaseous exchange and the transpiration. Clear? Clear this part? Okay. Now we are talking about how that lenticell get formed. Obviously हमने ये तो डिस्कस किया, lenticels present है, dot present on the stem, root and the fruit also. So we are talking about how that lenticell, how that small pore create on root region, create on the stem region. So next part we are talking about formation of lenticell, how they get formed. ठीक है? Formation of lenticell. So just listen carefully. We are taking some flowchart. Start from the Cork cambium. You know very well what is meaning of the Cork cambium. Okay. Such Cork cambium basically they cut from outer side. They cut from the outer side. And now what happened? After the cutting, now there is a formation of the some thin cells. Formation of some thick cell. and that thick cell also called as the cork cell, which is a dead. Okay, and as well as there is a formation of some thin cell. So cork cambium, last class also we discussed, they cut from the outer side and they form a phellum, and phellum also called as the cork cell. Cork cell, basically these are the dead cell, deposition of the tannin, deposition of the suberin material. So ultimately they are the quite thick. So when cork cell they cut toward the outer side, now there is a formation of the thick cell, that is cork cell, as well as there are formation of the thin cell. So we have to focus on thin cell. So now what happened? Just that thin cell which is get formed. So they create pressure. They create pressure on epidermis. Whatever the small thin cells are present, that thin cell they create pressure on epidermis, and finally on the epidermis there is a formation of the lenticell after the breakdown. So epidermis, due to pressure, it will be get break, and after the break, now final stage, there is a formation of small pore on stem on root area, and that cell nothing but we are talking about the lenticell. So just check what is the condition is present. We start from the cork cambium. Cork cambium they cut toward the outer side. Some cells thick will be formed, some cells thin will be formed. Thick cell simply we are talking about they are cork cell, उसका तो कोई problem ही नहीं है. But now what happened? Some thin cells, which is obviously living cells, they exert pressure, they create pressure on the epidermis. So now what happened? Where the thin cells are present, they exert pressure toward the outer side, they apply pressure, and due to that pressure, now slowly outermost epidermis layer they get break, and after the breakdown, now there is a formation of this small pore, and that pore nothing but these are called as the lenticell. Got it? Clear this point? How the lenticell they get formed? हम diagram भी देखेंगे इसका, don't worry, because we have to connect vascular cambium with cork cambium. Clear this part? Hello, Mangesh. Clear? Okay. Now quickly we are taking diagrammatic representation to represent cork cell, secondary cortex, cork cambium, as well as the lenticell. ठीक है, चलो. Now we are talking about simple diagrammatic representation. तो sequence याद रखना है. So first of all we draw cork cambium. क्या हो रहा है ये? हाँ. So this color we are talking about, these are the cell that is cork cambium, also called as the phellogen. Cork cambium they cut toward the inner side, and you know very well, once they cut toward the inner side, now there is a formation of which cell? Cork cambium cut toward the inner side. Now there is a formation of the which cell? Quickly tell me. Which cell? Cork cambium cut toward the inner side. Yes, it is a phelloderm, and phelloderm also called as secondary cortex. Very good. Okay. Now just we discuss cork cell they cut toward the outer side, and once they cut toward the outer side, two different types of the cell get formed. Thick cell called as the cork cell and the thin cell. Okay. So just quickly we draw. I am taking some outline. इनका थोड़ा सा हमें डोम शेप में ड्रॉ करना है, like this. So we are talking about now there is a formation of the another cell towards the outer side. So now there is a presence of thick cells like this. Cork cell, basically they are the brick shape. We try to draw a brick. तो कुछ इस तरह से cells हैं. Okay, and now this one, whatever the cells are given, this one, ये हमारे cells होंगे, that is called as the cork cell, which is the dead है plus ये thick भी है, thick plus dead. ठीक है, simple funda, cork cell. Now we are talking about thin cell which is lie at the center. तो यहाँ पे present होंगे. Now there is a presence of some thin cells. This one, and now what happened? This thin cell, they exert pressure toward the outer side. So just see. Now there is a presence of epidermis like this. These are the outermost epidermis. Okay. And now what happened? Due to this central cell, these are the living cell. Due to their activity, now that epidermis, it will be get break. Epidermis क्या होगा? Break होगा. And due to that breakage, now there is a formation of a small pore on epidermis like this. And that small pore, simply it is called as lenticell, which is always open, responsible for the gaseous exchange. And whatever the green colors are shown, this one, these are the thin cells, which is a living. Obvious है, if cells are living means they are showing continuously growth. अंदर तो कोई जगह ही नहीं है because already cork cambium is present. So now what happened? They exert pressure toward the outer side. And now due to addition of the new cells, epidermis, they get break. Can you see? Entire layer it is covered with the blue color. That is the epidermis layer. But at the place where living cells are present, that epidermis, it will be get break, and now there is a formation of the small pore, and that small pore, nothing but these are called as the lenticell. And please remember, lenticell, basically they are having a specific type of this shape. Why it is called as the lenti? Because shape of this pore, it is a lens shape. So on the basis of that, simply it is called as lenticell, which is responsible for gaseous exchange, major function. Second function, responsible for the transpiration. They always open because stomata close and open hota hai. Stomata open during the daytime and stomata close during the night. But lenticells, these are the specific type of the pore which is always open, day, night. कोई भी condition होने दो, they are always and always open. Got it? Clear? If you see NCRT diagram थोड़ा सा complicated है, but try to understand, meaning same है. I think these are given at the last page something. चलो बताओ जल्दी. Done? Padma, done. Spruha. Okay. So just try to understand one thing. ये तो पार्ट हो गया. These are the totally related to cork cambium, and cork cambium they present toward the outer side. So now what happen? यहाँ पे क्या present है? Secondary cortex. Exactly below the secondary cortex now यहाँ पे present होगा secondary phloem. Because we are talking about which growth pattern? Secondary growth. Exactly below the secondary phloem, you know very well. Now there is a presence of the vascular cambium. Whatever the other cells are present, all those other cells they undergo the process of degradation. Ha, Heram, true. Same diagram. Okay. So vascular ये जो complementary cells है ना, that complementary cells nothing but these are also called as the thin cell. Okay. If you want, you can write down this point. Thin cells which is a living, these are also called as the complementary cells. Hmm. Thin cells, living cells, they are also called as the complementary. Because now what happened? Number of that complementary, number of that thin cell, living cell, they are continuously adding, and due to that condition, they exert pressure on the epidermis. Epidermis break hoga, formation of the lens-shaped pore, that is called as the lenticell. Done. चलो. So we are talking about see the sequence, किस तरह से arrangement है. Here the cork cambium is present. Exactly below the cork cambium, secondary cortex hoga. Exactly below the secondary cortex, now the secondary phloem present. Below the secondary phloem, vascular cambium is present. Now below the vascular cambium, now there is a presence of the secondary xylem. So these are the exactly arrangement from the outer to inner at adult stage, at the lateral growth. Okay. So whatever jitne bhi cells honge like endodermis or the general cortex, they are showing they undergo the crush because secondary phloem number get increased. Cork cambium showing activity, secondary xylem also they are showing activity. So whatever the living cells are there like teeth, like lateral cells, like endodermis, all those cells they undergo the crushing, means they all crushed, and finally at the place of secondary organ or secondary tissue they get developed. So these are the exactly position. See कहाँ पे क्या changes हो रहे हैं. Outermost cork cell, now cork cambium, then secondary cortex, then secondary phloem, then secondary cambium, and finally secondary xylem. Clear? ठीक है. Done. अरे पापा ये secondary xylem क्या है? ऋषिकेश, secondary xylem means what? Nothing but यहाँ इसके यहाँ पे present होगा sapwood, and now finally presence of the heartwood. That's it. And you know very well heartwood exactly these are the center of stem or center of a plant. Hmm? चलो. Yeah. So any doubt from the lenticels? Any doubt? Done. Chalo. Next part we are talking about bark. Bark basically these are the one technical concept. So next part we are talking about region of bark. You know very well whenever we are talking about the bark area it means that all the tissue which is present toward the outer side. But we are talking about bark basically these are the one technical term where inside the bark we have to consider secondary phloem, as well as we have to consider region of periderm. And you know very well, inside the periderm, phloem bhi hoga. Again, they consist of the phloem as well as they consist of the phloderm. So collectively, these are called as the which derm? That is periderm. So please remember, whenever we are talking about the bark it is consist of the secondary phloem as well as they consist of the periderm, where it is consist of the phloem as well as the phloem as well as the phloderm. Got it? Okay. And now we are taking the a common example, best example for the bark. You know very well, during this treatment for the corona, एक drug use किया जाता था, hydroxyquinone, which is an anti-malarial drug, right? So that hydroxyquinone, that drug, which is an anti-malarial drug, used frequently, means frequently they are used for the during for the corona patient also. So that drug, it is obtained from the bark region. So we are talking about one plant, name of the plant, that is cinchona plant. Name of the plant, that is cinchona plant. From the cinchona plant bark, we have to remove which bark? Bark. We obtain one drug. And name of the drug, basically these are the anti-malarial drug. We obtain one anti-malarial drug, and name of the drug, quinone. Quinine drug.
Excuse me, ma'am.
Hydroxyquinone actually, listen, hydroxyquinone, basically these are the one technical name, but commonly when whatever the bark region is there, from the cinchona plant bark we obtain one anti-malarial drug, and that drug nothing but that is quinine drug. That's it. हाँ पूछो. हाँ,
Ma'am, can you
Ma'am, could you please
once again?
explain or scroll up.
Yes, ma'am.
Scroll up.
Ma'am, can you explain it once again?
हाँ. Listen. Bark means what? You know very well. Plant, it is having the bark. Bark means what? Which is the outermost dead tissue, which can be easily removed, right? So bark, basically these are the one technical term. Technical term means what? It is consist of the secondary phloem, and you know very well, secondary phloem they obtain from the vascular cambium, right? Another one, major region, major part we obtain from bark consists the major part from the periderm, and periderm is what? Cork cambium, cork cell, and the secondary cortex. So phloem nothing but cork cell, phloem nothing but cork cambium, and the phloderm, that is secondary cortex. So cork cell, cambium, secondary cortex plus secondary phloem together matlab from secondary phloem, all the tissue which is present outside from secondary phloem, all those entire region is known as a bark. Secondary phloem के outside जितने भी cells है, means secondary cortex, now the cork cambium, now the cork cells, all those regions they collectively called as the bark. Got it? Clear? And we are taking the one plant where bark is used as a drug. Name of the drug, quinine drug, which is one anti-malarial drug, and for the corona treatment also such drug already used initially. ठीक है?
Yeah, understood, ma'am. Thank you.
Yeah, okay. यहाँ से क्वेश्चन बनेगा डियर. One question from this part. Bark. चलो डन. Done. Chalo. So quickly, last point, we are talking about some difference between the vascular cambium in root and vascular cambium in case of the stem. Then we end this chapter. Next part. So quickly, we are talking about just 2 points. What is the exactly difference between vascular cambium in case of the root system and as well as the vascular cambium in case of the stem. दोनों देख रहे ठीक है because already we discussed in detail stem but now we are taking only some information with respect to root. Normally what happened in case of the root, whatever the vascular cambium is present, initially they have the wave structure. Waves are only. And now slowly they converted into circular one. And finally they converted into circular structure. So normally what happened? Waves means what? It's not circular. Exactly it is कुछ इस तरह structure होगा. Like this. So these are the vascular cambium which is present in case of the root initially. But now what happened? After some time, now it is converted into circular vascular cambium like this. Now when you compare this point with vascular cambium in case of this stem, so हमने देखा था always and always they are present in a circular manner because in detail we discuss how that vascular cambium they get formed in dicot stem. Remember now? Origin कैसे था? Intravascular cambium they bind with the intervascular cambium. Now there is a formation means intrafascicular cambium bind with the interfascicular cambium. After the joining, they form a one ring shape, circular shape that is called as the vascular cambium. So such kind of this vascular cambium, they are always and always circular in case of this stem. Now another point, if you talk about the origin, so you know very well, vascular cambium basically they partly primary as well as secondary in origin. Partly primary as well as the secondary. So I hope you remember हमने discuss किया था. There is a presence of the intrafascicular cambium, which is a primary part. Now they join with interfascicular cambium. Interfascicular cambium, and after the joining, now there is a formation of one ring shaped cambium that is called as the vascular cambium. Circular shape vascular cambium. So that's why we are taking one word, partly primary and the partly secondary means 50-50%. But please remember when you compare vascular cambium in case of the root, dicot root, so always and always they are secondary in origin. No primary. Completely they are secondary in origin. Okay? Yeah. Now, next point, I think you remember we discussed annual ring formation under the vascular cambium. So you know very well if at the whole year, if constant environment is present, temperature constant, rainfall constant, so that time vascular cambium activity is more and now there is a no formation of the spring wood, means only one type of the wood get formed because favorable. So there is a no variation in wood formation. But once we are talking about the temperate zone, temperate region, these are the favorable for the formation of the annual ring because variation with respect to favorable and the unfavorable environment. तुमने देखा था during the favorable spring would get formed, during the unfavorable autumn wood will be formed. So now what happened that spring wood and the autumn wood collectively they represent as a annual ring. So please remember this point. Vascular cambium, when you study in case of the stem region, that time they can form a annual ring in temperate zone. They can form annual ring in temperate zone, in temperate region. But in case of the vascular cambium, with respect to root, now there is a no formation of annual ring. Can you tell me why? There is a no formation of the annual ring. Anyone? Can you tell me why there is a no formation of the annual ring? Vascular cambium and stain, वहां पे तो हो रहा है, but when you are talking about vascular cambium in case of the root, there is a no formation of the annual ring. Why? Tell me, think ना बाबा, root कहाँ पे होता है? Vascular cambium associated with root. Root कहाँ पे present होगा? Because there is a favorable condition throughout the year. Yash, Nare. Roots are always have the supply of water. ठीक है, correct है, partially correct है. Listen, no formation of the annual ring. Why? Because root कहाँ पे present होंगे? Root present under the soil, and inside the soil, under the soil, constant environment is present. मतलब water भी present होगा and temperature also constant. But now what happened? Vascular cambium, which is present at the stem region, means that exposed to external environment, and if external environment get changed, ultimately they can directly affect on growth of vascular cambium. So simple reason is why there is a no formation of annual ring in case of the vascular cambium in root, because root present or such vascular cambium they present under soil where constant environmental condition is present. That's it, simple. Yash, see, when we are talking about dicot, हम dicot के बारे में बात करें. So, and as well as we are talking about the lateral growth, it means that whatever the roots are there, that roots they deeply situated inside the soil. They are not present on the surface. हाँ, हम ऐसे कह सकते हैं कि whatever the roots are present near to the soil surface, might be possible they get affected. हो सकता है. But we are considering that is vascular cambium which is present inside the root area, they present in deep region. So ultimately वहां पे तो constant रहेगा ना environment. Okay, because why we are talking about the temperate zone? Because extremely heavy winter is there, snowfall is there. Hmm? Okay, yeah. So these are the some basic information with respect to vascular cambium in case of the root and the vascular cambium in case of the stem. And with
Excuse
this,
me, ma'am.
हाँ Sania,
What is written after soil?
soil where constant environmental condition. So with this, successfully, happily, we cover the chapter anatomy of flowering plant. That's it. So any doubt? Any doubt? If you want to listen this chapter again, so after 15 or after 1 month, you can join with coaching batch. After 1 month, next month. So you will get notification related to meeting. So you can join.
Ma'am, what before constant?
Constant कहाँ है? Constant environmental condition.
Ma'am, and before
Present. Because under soil where constant environmental condition present.
Okay, ma'am. Thank you.
ठीक है? Yeah. So any doubt from this chapter?
कर दो और मचरा ऊपर
हाँ herum, hydroxychloroquinone, these are the different derivatives of quinone drug. It derivatives है. That is also true. It's derivatives, different form. Some chemical formula they get changed. That's it. Chalo, should I start with cell? Next chapter, cell cycle and cell division. Chalo. So next part, very simple, very easy chapter and quite interesting chapter, and according to neat point of view, important chapter, cell cycle and cell division. Hi, it's true, Baba, Hiram. It's true. Okay, chalo. So next part, we are talking about chapter cell cycle and cell division. Cell cycle and cell division. Normally, what is meaning of the cell cycle? Cell cycle, nothing but we are talking about some orderly series get changes. मतलब cell present है, cell now they present in G1 phase, now they entered into S phase, G2 phase, M phase. So something changes will be occur, but in a particular sequence. So cell cycle, nothing but it is an orderly. Simple definition. It is an orderly series of changes. Series of changes that occur in a cell. Series of changes. Normally, what happens during the developmental stages when we are talking about the cell cycle? Normally, there is a presence of any mother cell or parent cell. So, suppose there is a presence of one mother cell. Okay. Now, this mother cell, it is undergo the process of duplication. So, they can duplicate their genome, they duplicate their all material, they can duplicate their cell organelles, and after the duplication, they form two daughter cells. So, finally, there is a formation of the two daughter cells. Okay. So, what is meaning of the process? Cell cycle means what? These are the some series of changes which is occur, and due to that series of changes from mother cell there is a formation of the two daughter cells. So what kind of the changes will be occur? How the DNA get double, at which phase it is get double, cell organelles duplication or RNA, protein, enzyme duplication. हमने organelles देखे थे, in case of cell chapter we'll discuss the different organelles. We try to connect cell chapter with this cell cycle cell division. So in this chapter we'll try to understand from one cell how we obtain two cells. Okay? Now what happened? It is a simple definition for the cell. Next concept we are talking about generation time. Next part we are talking about the generation time. Simple definition for the generation time, that is total time which is required to complete one cell cycle. So generation time nothing but that is the time period, total time required to complete one cell cycle. ठीक है, to complete one cell cycle. So obviously ये क्या part हुआ? This part nothing but we are talking about these are the cell cycle, one cell cycle. From one mother cell we obtain the two daughter cell. So how that two daughter cell get formed, how much time it is required, that time period simply it is called as the generation time. Now to try to understand generation time we are taking few examples. For example, first of all we are talking about in case of the human. हमारी बात कर रहे हैं. Basically human, we have the genome size approximately 2 meter, or in base pair 6.6 into 10 raised to 9 base pair. So human it has very high genome. Cell size भी large है. Genome size also large. So as compared to bacteria, as compared to unicellular organism, human required more time to complete cell cycle, single cell cycle. And in case of the human, 24 hours required for generation time. These are the time, basically 24 hours. These are the generation time in case of the human. And just we discuss generation time, nothing but the time which is required to complete one cell cycle. From one cell we obtain the two cell. कितना time चाहिए हमें? 24 hours. Okay, human. Another one, we are taking the example of the yeast, Saccharomyces cerevisiae. You know very well, yeast basically these are the example for unicellular eukaryotes. This example for the unicellular eukaryote, and genome size is quite small, but still it is required 90 minutes. Generation time, basically these are the 90 minutes, means 90 minutes is required from obtain two daughter cell from one mother cell. And finally we are talking about the Cinderella, that is E. coli bacteria. Cinderella, I called her Cinderella because E. coli fantastic bacteria hai. Everywhere it is present. Lots of biotechnology experiment it is done with the help of the E. coli. जितने भी experiments हुए related to bacteria, maximum experiment are done with the help of the E. coli. So personally I called E. coli as a Cinderella. So Cinderella having the very small body, genome size also very small, 6.2 into 10 to 6 base pair. So normally what happen, whatever the generation time is required, the generation time is quite less. So E. coli bacteria generation time it is only 60 minutes because genome size is small, and you know very well we are talking about the E. coli bacteria, Escherichia coli bacteria, and bacteria we can't see by the naked eye. So body always small, body small, genome size small, so they require quite less time. Approximately 60 minutes are required for E. coli bacteria. Again I'm telling you, bacteria to bacteria vary hoga. If I'm talking about the bacillus bacteria, lactobacillus which is present in curd, or normal bacillus, they require approximately 120 minutes. So it's depend upon the genome size of bacteria. So for E. coli, generation time is 60 minutes. Got it? Clear?
Clear, ma'am.
Clear? Clear now? हेरम्प और यश के मौज है अभी. They already covered all this part. ठीक है, चलो. Next part we are talking about cell cycle phases. Cell cycle. Basically, cell cycle in this chapter will study into two parts. Number one, I phase, and I phase also called as the interphase. I phase also called as the I stand for the interphase. I phase also called as resting phase. These are also called as the resting phase. And why it is called as the resting phase? Because these are the phase where no cell division. यहाँ पे cell division नहीं होता. Resting phase के बाद we are talking about I phase also called as the preparatory phase. These are also called as the preparatory phase. And why we are talking about the preparatory phase? Because these are the phase where synthesis of DNA, RNA, protein, enzyme, cell organelles, ATP, everything can be synthesized. So I stand for the interphase. These are also called as the resting phase because यहाँ पे cell division नहीं होगा. Only cell synthesize all material before division. So that's why it is also called as the preparatory phase. And why these are called as the preparatory phase? Because these are the phase where synthesis of different material like synthesis of DNA, synthesis of RNA, synthesis of enzymes, synthesis of proteins, synthesis of energy in the form of ATP, and as well as there is a synthesis of different cell organelles, because from one mother cell we have to obtain two daughter cells. So whatever the amount is present in one cell, each and every amount, चाहे वो न्यूक्लियोप्लाज्म का अमाउंट होने दो, amount of DNA, amount of protein, amount of enzyme, cell organelles, सब कुछ double होगा, because from one cell we obtain two daughter cells, and each and every material they equally distributed into daughter cell. So ye I phase A, which is a quite longest phase, and now such phase, these are the phase where different part will be synthesized. So preparatory phase A, preparation of DNA, RNA, enzyme, protein, ATP, as well as the cell organelles. Okay, so ये था first part with respect to cell cycle interphase, which is a quite longer phase. Done. Chalo. Done. Okay. Now we are talking about the next phase, which is a very small phase, M phase, capital M. And the M phase, they stand for mitotic phase. M stand for mitotic. I stand for interphase. And please remember, these are the phase, mitotic phase, where cell division will be occur. That's so quickly we are taking the difference between the I phase and M phase. Done. In this chapter, we'll discuss this in detail. Is what kind of the changes will be occur, what kind of the event will be occur in interphases, and what kind of the changes event occur in mitotic phase. So we'll explain each and everything in detail. Hello. Yeah. So please divide your page into two sections. Now we are taking the difference between the I stand for the interphase and M stand for mitotic phase. So first of all, we are talking about the I phase compared with M phase. Basically, I phase just we discuss, these are the preparatory phase. Obviously, preparation kar rahe hain. Before cell division, before formation of the two daughter cell, now they get prepared. So obviously, according to time, it's quite longer phase. It's quite longer phase. And if I'm talking about in percentage, approximately 95% time out of cell cycle. Suppose कि अगर 24 hours की cycle है, suppose 24 hours. So out of that 24 hours, maximum time, that is 95% time, it is required for the I phase, because these are the phase which is a quite required longer duration. Longer phase, or you can say they required the quite longer duration. Longer duration, because they required how much time? Approximately 95% out of 24 hours, they required more time. When you compare M phase basically it is very dramatic phase. बहुत ज़्यादा drama हो रहा है. In the sense, it's very shorter. First of all, it's very shorter duration it's required, and out of 24 hours, just 5% time it is required to complete the M phase. So if I tell you kitna time lagega for the human, so this longer duration means 95% means you can consider approximately 22 to 23 hours are required in case of the human, because human generation time, human cell cycle require 24 hours for the I phase. But When we consider 5%, it means that hardly they require 2 to 1 hours in case of the human, because it's very shorter but lots of drama happening with M phase. हम आगे देखेंगे कंडेंसेशन, डीकंडेंसेशन, क्रोमोसोम मूवमेंट, न्यूक्लियर मेम्ब्रेन, सब कुछ डिसअपियर होता है, रीअपियर होता है, लॉट्स ऑफ मूवमेंट्स, लॉट्स ऑफ रैपिड मूवमेंट्स आर ऑकर ड्यूरिंग द एम फेज बट शॉर्टर. ठीक है. या second part we are talking about, already we discussed I phase, nothing but these are the preparatory phase where there is a synthesis of the cell organelles, synthesis of the enzyme, protein. So ultimately what happened, due to preparation, due to synthesis of the all material, they are such phase, it is responsible for increase in cellular structure. Size of the cell will increase, it will be showing the growth, multiplication of the different material. So now we are talking about, these are the phase where cellular structure will be increased. And why it is increased? Because these are the phase where synthesis of material will be occur. Different kind of the materials, they get synthesized. So obviously क्या होगा यहाँ पे, the cellular structure size, it will be increased. And now what happened, these are the phase, M phase, where division of cellular structure, whatever the material gets synthesized, ATP, enzyme, cell organelle, जो कुछ भी बनाया, everything. So everything or each and every material, each and every cellular structure, they divide into two daughter cells. So these are the phase where division of cellular structure, into daughter cell. Was it the M phase? Okay? And finally, along with material, you know very well, genetic material also they get double. So we are talking about whatever there is a presence of the genetic material, that is hereditary material, which is present in the form of DNA. So whatever the genetic material is present, that genetic material basically they present in a thread form. Genetic material formed a thread-like structure. Thread-like structure. And such thread-like structure, simply it is called as chromatin. It is called as chromatin, or simply you can say these are simply it is called as the chromatin fiber. In cell chapter we already covered this part. So quickly I'll draw the diagram. Now structure कैसे होगा? Chromatin, genetic material, hereditary material, like this. So they present in which form? They present in a chromatin fiber form, like this. But now what happened? Cell once committed to M phase. Now whatever the genetic material is present, all this genetic material they converted into chromosome. So genetic material, now they become highly condensed. They become highly condensed and finally they converted into chromosome structure. Chromosome, and you know very well, when you draw any chromosome structure, it is has the typical structure, presence of the centromere with P arm as well as Q arm. So these are the typical structure of the chromosome, which is a highly condensed. So यहाँ पे क्या हो रहा है? Normally chromatin fiber they present in a loose form, right? Chromatin fiber they present in a loose thread-like structure, but now slowly they convert it into chromosome structure, that is highly condensed structure. So chromatin fiber, loosely form of genetic material, loosely form of the hereditary material, they present during the I phase, but one cell committed to cell division. Now that time, slowly that thread-like structure, chromatin fiber, they converted into chromosome, which is a highly condensed structure. Done? So these are the exactly major difference with respect to I phase and M phase. Done? Padma? चलो हो गया. Should I continue? हेलो, सो गए क्या? हेलो, सानवी. ठीक है. So now these are the exactly difference with respect to I phase and the M phase. Now we are talking about the next part, cell cycle diagram with each and every phase in detail. So please remember, in our syllabus basically we have to study cell cycle in case of the human. जो कुछ भी हम part ले रहे हैं that is related to human. We are taking cell cycle in detail, and we are taking cell cycle in case of the human, or you can say in case of eukaryotic system. So normally what happens when we are talking about the cell cycle, few phases are included. For example, there is a presence of the G1 phase, followed by now there is a S phase. After that, now there is a presence of the G2 phase, and finally there is a presence of the M phase. So these are the exactly sequence for cell division: G1, S, G2, and M. So quickly, one by one, we'll discuss what is the exactly cell cycle and what kind of the event will be occur. So first of all, we try to draw a simple diagrammatic representation for cell cycle. So in this condition, suppose one section, this one basically G1, gap 1. Now we are talking about followed by S phase. So ये हो गया S. Now there is a followed by G2. So ये हो गया G2, and finally this entire phase basically it is called as the M phase. Finally we are talking about the M phase. M phase, it is divided, that is mitotic phase, cell division phase. It is divided into two types. M phase, it is divided into two types. Number one, karyokinesis. And what is meaning of the karyokinesis? Tell me. What's the meaning of karyokinesis? And the followed by number second, cytokinesis. What is meaning of karyokinesis? Tell me, anyone. Division of nucleus. Yes, nucleus division. And what is meaning of the cytokinesis? Very good. Karyokinesis nothing but nuclear division. And karyokinesis means what? Yes, Amod. Karyokinesis, cytokinesis nothing but that is cytoplasmic division. So exactly these are the difference between the karyokinesis, that is nuclear division, cytokinesis, that is cytoplasmic division. Now, when you talk about the karyokinesis, that is nuclear division again they divide into subphases. Number one, prophase, followed by metaphase, then followed by anaphase, and finally telophase. So listen carefully. We are talking about M phase. Just we discuss M phase. Basically, it is quite shorter phase, but lots of drama happening during this phase. Drama means what? बहुत सारे changes हो रहे हैं. Because under the M phase, they divide again into two types. Number one, karyokinesis. Number second, cytokinesis. Karyokinesis means what? Nuclear division. And nuclear division again they divide into four stages, four phases like prophase, meta, ana, and telo. And finally, cell undergo cytokinesis process. So quickly we draw those structure. यहाँ पे present होगा first of all prophase, metaphase, anaphase, telophase. So I just give the initial letter. यहाँ पे present होगा prophase. Now followed by meta, followed by ana, followed by telophase, and finally there is a presence of cytokinesis, that is cytoplasmic division. So just quickly check, we are talking about under the metaphase. पहले होगा G1, after the G1 now there is a S phase followed by the G2 phase. After cell entered into M phase divided into karyokinesis. During the karyokinesis पहला phase होगा pro, then meta, then ana, then telo. Please remember, you have to remember sequence because cell cycle definition, these are the series of changes. Okay, orderly change हो रहा है but in a specific sequence. So please remember, when you study cell cycle, you have to remember sequence. पहले होगा G1, then S, G2, and finally M. ठीक है? Under the nuclear division, first of all pro, then meta, then ana, and finally telophase. Chalo. Now quickly we are talking about which part it is included into I phase. So you start from the G1, then S phase, then up to G2 phase, this one. So all these phase it is included into I phase. So please remember, when we are talking about the interphase, हमने देखा these are the synthesis phase, preparatory phase, interphase, right? So during this interphase, now they included G1 plus they included S as well as they included G2. So G1 is G2, all these three, these are the part of I phase, these are the part of the interphase, and during the M phase, during the mitotic division, now there is a presence of the karyokinesis as well as there is a presence of the cytokinesis. So quickly complete the diagram carefully. Chalo, done. 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 Yeah. So any doubt from today's class? Any doubt from the next class? We start with the G1. Detail what kind of the changes will be occur. G1 is G2 sequentially. So any doubt?